So for the remainder of the processing here, I'm going to remove the lat and long so that we can see the graph a little more clearly. So I select lat long and unclick the box and say OK, right click the map and redraw. OK. Now we're going to want to look at the individual profiles to make sure that the data is in the correct range and that there are no anomalies that need to be corrected. So I can go to File, go to New, and make a profile view. And if I select a profile view, we see the actual profile of this particular line. And again, we know which line it is because it's selected in red. So we can look through the data. I can select a different line. One way to select go through the different lines is to open the browser. And I go to File, Browse, and this opens a browser control panel where I can go line by line and look at the data. We see the position in meters and in meters. And we can go through the individual profiles and make sure that the data is clean and correctly scaled and so forth. If we had, for instance, a particular anomaly that was of interest, we can set cursors on that profile, and then when we look on the the gridded map, the profile grid map, we can see where that anomaly is actually positioned. Continuing down through the different maps. Now here we see we have a spike of some sort, so we're going to zoom in on that. And we see that it is in fact a very short amount of time. It's in fact over just less than 1 meter. We can see that this actual anomaly or this spike is over a very brief period of time. So in order to cut it out of the data, we set our markers at both ends of the spike, and then we go to Edit and we go to Cut. And it gives us some options. Cut from the start of the line to the up marker. That's not correct. Cut part after up marker to the end of the line. No. Cut outside up and down markers. No. Or cut between up and down markers, which is correct. So we're going to cut out this spike which is sitting in between here. We just say OK, and now we see that it rescales and that small amount of data has been removed. Go through the rest of the lines, making sure that they are that there's no anomalies or other types of corrections needed. We can see in the profiles here both the red and the blue. The left and right magnetometers are shown by red and blue lines here. We have 56 lines in the file. And when we come to the end of the file, we now have observed every profile, made sure that it's clean data and that there are no spikes or other types of anomalies. One reason that we're looking at these profiles is that we want to get a feel for the variability. We want to make sure that we don't have any very noisy sections for some reason. We want to make sure that all the lines have basically the same amount of change, and that we can see that there are anomalies associated with each of the magnetometers separately. Okay, if we want to spread this out, and the way in which we control the individual the profile screen is to right click on it and go to Settings. And here we have the shrink to canvas button, which keeps it on the page. We can profile it along the X, along the Y, or along the profile. In this case, our lines are pretty much north-south, so along the Y looked fine, but normally we would want to profile them along the actual position of the line itself. So we would go along the profile. The T size is the magnetic size, or the Y-axis, and the X-axis is the number of meters. In this case, we set it to 1,000. We could set it to 500, which would make for a shorter map, and so forth, and we can see that. 1,000 is probably a better setting to be able to see the anomalies and get a sense of the data. Now that we've gone through the individual profiles and identified the geology and some of the anomalies, and the fact that we have both magnetometer files concurrently showing the geology and the profiles, now it's time to save the project. The project saves any edits that we've made to the data, and it allows us to reopen the file at that point in our project. So we go to File, Projects, Save As, and we can save it. Gives it an extension .MagPick, and I'm going to save this one as Demo Project, and it'll add a .MagPick. And now I can always get back to this point and open the project and see the data files in this position. We're now going to compute the gradient. So we must, in order to compute the gradient, we must close the profiles window. So it's important that we close that so that we don't have the individual profiles listed. So we go to Profiles, Compute Gradient. And this dialog box opens, and the first thing we have to answer is how far apart were the sensors. In this particular transverse gradiometer, the sensors were separated by 1.5 meter. So we put in 1.5 meter. Magnetic sensor number 1 channel is in number 1. Magnetic sensor number 2 channel is in number 2. And the vertical middle channel, which we're not sampling with a third magnetometer, we're computing the vertical gradient, so it's grayed out. We don't have that option in this particular dialog box. Alright, the gradient type will be pseudo-analytic signal. That means that we are computing the transverse gradient, we are computing the longitudinal gradient. From those two gradients we are computing the vertical gradient, and from that we are computing the pseudo-analytic signal, which will be put into a separate file that we can then open and contour. Okay, so I'm going to list this file name as Demo Gradient, and it'll be given the extension .dat.